Today we're going to talk about microbial growth, Chapter 9. Many things come into play when we're talking about the dynamics of prokaryotic growth. Remember, bacteria are incredibly diverse. Each species has specific nutritional needs. It has specific environmental needs. It needs to grow under certain conditions. Each species has their own requirements. In addition, when we're trying to grow these different prokaryotes in different types of media or on different types of media, they produce certain characteristic byproducts. For instance, if we do sugar fermentation, we look at how that bacteria will metabolize that lactose or that sucrose or that glucose. We look to see what they produce. Do they produce a lactic acid? Do they produce a gas? When a bacteria metabolizes their specific nutrients, they will produce these byproducts that we can visualize. We can take advantage of all of these things to identify bacteria phenotypically by what we can visualize. We have to be able to see it. We have to see the changes that it can make to the augers that we place them on or the mixtures of broths that we put them in. We can see how they grow. Do they grow aerobically? Do they grow anaerobically? Are they dispersed throughout the media? Are they wet when they grow? Are they dry when they grow? We can look at all kinds of different things phenotypically to see what that type of bacteria is and identify it. First, in order to begin our identification process, we have to have a pure culture. If we have a pure culture and we have worked an environmental sample down to one species of bacteria, then we can do all of these different things to it and identify it. But only 1% of all microorganisms can currently be grown in a pure culture. 1%. That means that 99% of microorganisms cannot be grown in a pure culture. So let's talk about those pure culture basics and some definitions that you will need to know. Being sterile means it is completely free of microbes. Aseptic technique is procedures that we use to minimize unintentional introduction of microorganisms. There are certain things that we do in the lab. We flame the loop until it's red hot to make sure that we are not adding anything to our samples that have been sitting on the loop, that have been in the air, that have landed on the loop, the leftovers that might be on the loop. We flame them until they're red hot. That is an aseptic technique. When we streak a streak plate, when we use a petri dish, we block the petri dish with the lid so that our breath does not introduce extra microbes to that plate. We use aseptic techniques by flaming the tubes that we culture from so that nothing extra is added to that tube. There are many things that we need to do aseptically so that we can make sure we're not introducing extra microorganisms to that culture. Now, when we talk about the different types of agar that we use, we can use many different types of agar. We use TSA agar in the lab. We use a blood agar in the lab. We use nutrient agar that has different types of nutrients to see if the bacteria likes it, or a real salty agar to see if it can grow in salt. These augars melt at 95 degrees Celsius and they solidify at 45 degrees Celsius. They are exceptional to be able to use in a pure manner, to incubate in the incubator, and to use them to identify bacteria. A streak plate starts with whatever type of auger you are using to identify the particular bacteria that you are looking at. What you want to do is grow a colony on this auger plate. And a colony is a group of cells that are all derived from a single cell. Or it's a single colony that's derived from just a few cells. Remember when we did the streak plate, we got smaller and smaller and smaller with our sample, thinner and thinner and thinner. We thinned it down until we got to that pure colony that was derived from that larger sample. That is how we work with pure cultures. Microbial growth is an increase in the number of cells, not the size of the cells, but how many there actually are. There are also many things that affect microbial growth, and we are going to look at that throughout this chapter. Temperature can affect the growth of microbes. Some like it really hot, some like it really cold. The pH, some can live in acid. Osmotic pressure has to do with the concentration of salt or the concentration of sugar. It can change the osmotic pressure. And some bacteria love salt and some do not. We will also look at the different chemical requirements of bacteria. What do they need to grow? Do they need carbon? Do they need nitrogen? What types of things can influence their growth? Prokaryotes have specific temperature ranges that they like to grow in. They have a minimal growth temperature where it has to be at least a certain temperature for it to grow. They have an optimum growth temperature, their favorite temperature in order to grow. And they have a maximum growth temperature. These are all influences on the growth of bacteria, and it has to do with the disruption in their cell membranes. Is it too hot for it to grow in? Do the cell membranes liquefy? Is it too cold for that particular microbe to grow in? Does the cell membrane become too stiff and too rigid to grow? We need to look at the different types of temperatures that bacteria and other prokaryotes like archaea like to grow in. It's very, very important to understanding and being able to look at the characteristics so that we can identify them. From this diagram, you can compare psychrophiles and the temperatures that they love to the temperatures that maybe a hyperthermophile loves. You can see that the psychrophiles love it to be colder, and the hyperthermophiles on the opposite end love it to be in a much higher temperature. We will talk about these individually on the next slide, and you'll need to pay attention to these temperature ranges. You also can see that the curves are skewed towards the optimum temperature. There is an optimum temperature where bacteria are the happiest. The skewing of this growth curve is thought to reflect that rapid denaturation of the proteins that are in the cell membranes. As the temperature rises past that optimum, you can see that it really drops pretty rapidly. That growth curve is pretty steep. And so you can tell that there is this optimum temperature and then this rapid decline once you get past that optimum temperature. When we look at these temperature ranges, you need to be aware of which bacteria prefer which types of temperatures. Do they prefer it to be cold, or do they prefer it to be really hot? Or maybe they're happy being in our body temperature. Most common microbes that cause infections are mesophiles that prefer our body temperature, 37 degrees Celsius. When we look at the psychrophiles, phile or philic means they love a certain type of temperature. They will be on the lower end for the psychrophiles. Psychrophiles can live in temperatures of negative 10 degrees Celsius to 20 degrees Celsius. That's extremely cold. Psychrotrophs mean they tolerate the temperature. Yeah, they like those colder temperatures, 0 degrees Celsius to 30 degrees Celsius. They like it pretty cold, but not as cold as a psychrophile. Think about your refrigerator. We use that refrigerator to slow the growth of microbes, to prevent food spoilage. You hope that those microbes in your food are not psychrophiles because you don't want them to like the cold. You are slowing down their growth. So those microbes in your food would be mesophiles or thermophiles. If they were psychrophiles, they would flourish in your refrigerator. They would really grow. So the idea of a refrigerator is to slow down the spoilage of food, to slow down the growth of bacteria because they have a certain temperature range that they like. So we talked about mesophiles and how their optimum is our body temperature, 37 degrees Celsius. Thermophiles prefer it to be warmer, thermo. Think about hot. Thermophiles like it to be 40 degrees Celsius to 70 degrees Celsius. That's pretty warm. Think about composters. Composters can heat up that compost pile. They like it hot and they really grow in a compost pile. Those are thermophiles. You can use thermophiles in industrial applications. We can use the enzymes from these hot, heat-loving, high-temperature-loving bacteria. Hyperthermophiles love extreme heats. They like it to be 65 degrees Celsius to 110 degrees Celsius. That's incredibly warm. That would be in deep sea thermal vents or in hot springs like at Yellowstone. Those type of microbes that can live in these extremely heated conditions, those are the hyperthermophiles. This is an image of something that we can refer to as strawberry snow or watermelon snow. It is actually a microbe that loves to be cold, and it grows in these really cold, weirdly changing conditions. But if you ever see colored snow like this, it looks pretty inviting. It looks like a snow cone or something like that, but we would never ever want to eat any colored snow, well, for many reasons, but different colored snows, like the red colored snows like this, you know that they are colored this way because there's a certain type of psychrophile that is growing in this very cold environment. This is an example of a deep sea thermal vent, like an underwater volcano, and it's extremely hot. So the type of prokaryotes that would like to live in these deep sea thermal vents that have these extremely high temperatures would be a hyperthermophile. Remember, you're looking at the words and you're trying to think about what type of bacteria is that? What type of temperature does that bacteria love to be in? Make sure that when you are looking at those words that you can relate them to the temperatures. So again, bacteria, prokaryotes, they have specific temperature ranges that they prefer, that they can survive in. They have a minimum, a maximum, and an optimum. That minimum survivable temperature has to do with the enzymes and that membrane fluidity. Is it too hot that membrane becomes loose and liquidy? Is it too cold that it becomes too solid, too structured, too tight for that prokaryote to survive? They have that maximum survivable temperature, and that's due to the enzymes denaturing. So you need to understand what causes that minimum survivable temperature and why there is a maximum survivable temperature. Prokaryotes also have specific pHs that they prefer, just like temperatures, and they have these ranges. Most bacteria grow between pH 6.5 and 7.5. But if you look at molds and yeasts, their preferred pH ranges are like 5 to 6. But then remember, there are acidophiles, and there's that phile word again, philic, it loves. So acidophiles grow in extremely low pH ranges, like the pH of your stomach. They can grow in a pH of 2 or a pH of 1. We can also capitalize on this pH requirement of prokaryotes. When we think about infections and trying to understand how someone has a wound and it develops an infection, there has been research done that has been trying to capitalize on the pH level that these bacteria prefer. So remember, we talked about how bacteria produce byproducts when they are in their environments, when they are using nutrients, when they are growing. If we can somehow capitalize on their pH requirement and the acidic byproducts perhaps that they produce when they are growing, we can use something to detect this level of these byproducts, this level of acid that might be produced from a bacteria causing an infection. And it's really cool. In 2010, researchers created a bandage that can indicate an infection based on detecting the pH changes, based on whether the bacteria changes it to an alkaline environment or an acidic environment, based on the bacterial byproducts that these bacteria can produce in their environments. It's really exciting research, and hopefully they will expand upon this. We also talked about how osmotic pressure can influence the growth of prokaryotes. Hypertonic environments cause plasmolysis. What that means is it's a high sugar content or a high salt content. The concentrations of sugar or salt are very high, and because they're high, they can create a high osmotic pressure. Now, there are extreme or obligate, which means they require this high concentration. There are extreme or obligate halophiles. They require a high osmotic pressure. They need it to have this high salt concentration or this high sugar concentration in order to live and be at their optimum. Facultative halophiles tolerate. They do not require a high osmotic pressure, but they like it. So facultative means they tolerate something. A prokaryote can tolerate something if it's facultative. Obligate always means that whatever it is, a prokaryote, a virus, whatever it is, requires a certain thing. Obligate means it's a requirement. So extreme or obligate halophiles require that high salt or sugar concentration that produces that high osmotic pressure. Staphylococcus is one of those types of bacteria that really can thrive and grow in a high salt concentration. If you think about our skin and our sweat, which can be really salty, you can think about that environment having a high salt concentration and high osmotic pressure. So the Staphylococcus would really enjoy our sweaty, salty type of skin environment for them to grow in. Let's look at some review questions. Of the total microbes in nature, we can grow 100%, 99%, or 1%. The answer is C, 1%. We can only grow 1% of all microorganisms in nature in a pure culture. That's really low. That means that 99% of the microorganisms in nature cannot be grown. So if we thought about how we could grow 6 microorganisms, that means basically if we round up, we cannot grow 600 microorganisms. 99% of that we do not grow. It's pretty amazing that we can only grow 1% of all microorganisms in a pure culture, considering the technology that we have, considering the research that we have done for so many years. It's pretty interesting that we can only grow and culture 1% of these microorganisms. Aseptic technique ensures A, specimens are sterile, B, specimens are pure and free of contamination as much as possible, C, the microbes have the necessary growth conditions to replicate. The answer is B. Specimens are pure and free of contamination as much as possible. We don't want to introduce extra things in there by having sloppy lab techniques. We want to do all of our techniques very cleanly so that we can grow a pure culture. We don't want it to be sterile, because if it was sterile, that means it's completely free of all microbes. We do not want A. What we want is B. Specimens are pure and free of contamination as much as possible with the use of our great aseptic technique. If you incubated bacteria on agar above 100 degrees Celsius, it would be a awesome thing, A, or B, a melted mess. The answer is B. If you incubated bacteria on agar above 100 degrees Celsius, it would be a melted mess. Remember, agars melt at 95 degrees Celsius. Your body temperature is 37 degrees Celsius. Most human microbes, the ones that cause infection, would then be A, psychrophiles, B, psychrotrophs, C, mesophiles, D, thermophiles. The answer is C, mesophiles. Most human microbes that cause infection that love our body temperature at 37 degrees Celsius, their optimum temperature is 37 degrees Celsius for a mesophile. Remember, the psychrophiles, they like the cold. They love it. Psychrotrophs, they tolerate the cold. Thermophiles, remember, love the heat. They like it when it's really hot. Your refrigerator would be an ideal environment for A, mesophiles, B, thermophiles, C, psychrotrophs. I bet you came up with C, psychrotrophs, because we've been talking a lot about it. Psychrotrophs like the cold. It starts with that psych and it ends in troph. That means they tolerate the cold. So your refrigerator would be an ideal environment for a psychrotroph. It would grow in there. A thermophile would not, because it needs heat in order to grow. It needs heat for prokaryotic growth. Mesophiles, again, they just like it right in the middle. They like our body temperature right in the middle, and they still would not like the refrigerator because it's still too cold for them. They need it at 37 degrees optimum. Which of the following has the highest osmotic pressure? Is it A, a 1% NaCl solution? NaCl is salt. Is it B, 10% NaCl solution? Is it C, a 10% sugar solution? Or D, 15% NaCl solution? Now, both salt and sugar can increase osmotic pressure. And what you're looking for here to get the right answer is the highest number. The highest percentage would create the highest osmotic pressure. So the answer would be D. 15% NaCl salty solution would have the highest osmotic pressure. So whenever you see a question like this, you're taking that highest percentage, that highest number, and because it is the highest number, it will also have the highest osmotic pressure. So again, the answer is D. Osmotic pressure damages cells by A, destroying the peptidoglycan, B, disrupting osmosis, C, poking holes in the plasma membrane, D, destroying the microbe's DNA. Well, you can kind of look for the similar words. If you weren't quite sure what osmosis was, you could still see that osmotic pressure has that similar type of word in there. You can also look at this question by ruling things out. We haven't talked about peptidoglycan when it comes to osmotic pressure. And we haven't really talked about poking holes in the plasma membrane. That's something that alcohol might do to a gram-negative bacteria. So it doesn't have something to do with osmotic pressure. And we have not talked about the destruction of DNA when we've talked about osmotic pressure. So if you didn't really know what the word meant, you could look at a question and rule out the things that you know are wrong. So ultimately, the answer for this question is B. It disrupts osmosis. What happens when you have a high osmotic concentration or a low osmotic concentration? It affects how the water goes in or out of a cell. It shrinks or swells a cell. And so osmotic pressure disrupts this movement of the fluid in and out. And that's what osmosis is. Osmotic pressure and the changes in the concentrations of a solute disrupts how osmosis occurs, how it flows in or how it flows out. So the answer is B. Disrupting osmosis occurs from osmotic pressure changes, and it damages the cells. We also mentioned that each type of microbe, each prokaryote has different types of requirements, different types of chemical requirements for their nutrients. Many prokaryotes require carbon. Carbon, just like how you require carbohydrates for your nutrients. Some prokaryotes require this carbon, and it's an energy source for them. Chemoheterotrophs use organic carbon sources. They use them for their energy. Autotrophs use carbon dioxide. In addition to carbon needs, sometimes prokaryotes have other types of chemical needs. When you look at this slide, there's a lot of information here, but the key points that you should be able to identify are that when you look at nitrogen and sulfur, both of them have to do with amino acids. They both have to do with proteins. Most bacteria can decompose proteins. So nitrogen and sulfur are involved in protein synthesis. Some prokaryotes need to have the chemicals nitrogen and sulfur in order to have the key components for protein synthesis. Also, you can look at phosphorus, and you can kind of remember if you've had A&P or if you've had biology, ATP might stand out to you. P is that phosphorus. So ATP, remember, is an energy component. So some prokaryotes need phosphorus for ATP. They also need it for their membranes. Think about phospholipid bilayers. For their membranes, they would use phosphorus. Some prokaryotes also have trace elements that they need. These can be inorganic or organic. So trace elements are inorganic elements that are required in tiny amounts. They're usually like cofactors, enzyme cofactors. Organic growth factors are organic compounds obtained from the environment, like extra vitamins they might need, or amino acids, or purines, or pyrimidines. There are certain things that they find organically that helps them in a certain way. It's just an extra nutritional need that they have. Oxygen requirements are very important to understand when we talk about microbial growth. For identifying bacteria, it can really point us in the right direction if it's an aerobe or an anaerobe, if it requires something or it doesn't want something. Remember that obligate word means that it requires something. Facultative means that it tolerates something. So let's look at oxygen requirements on the next couple of slides. And the next slide especially, I think, will help you. But let's look at the definitions here. Obligate aerobes means that it requires oxygen. It has to have oxygen. It is something that grows aerobically. It will not grow if there is no oxygen present. Facultative anaerobes. Now this definition is a little bit complicated. Facultative anaerobes have both aerobic and anaerobic growth, but it grows best in oxygen. So you would think with the word anaerobe, which means without oxygen, that it would grow best in a condition that's without oxygen, but that's not true for a facultative anaerobe. It grows both aerobically and anaerobically, but best in oxygen. But an obligate anaerobe can't have any oxygen at all. It will not grow when oxygen is present. Obligate, remember, is requires. Anaerobe means without oxygen. So, obligate anaerobes only have anaerobic growth. No growth will occur in the presence of oxygen. Now, aerotolerant anaerobes, they survive in the presence of oxygen. They tolerate it. Remember that tolerant means they're okay with it. Microaerophiles, only aerobic growth occurs and oxygen's required in really small concentrations, really low. This is one of my favorite images to help you understand those previous definitions. So if you look at A, it's an obligate aerobe. It requires oxygen. So if that bacteria were in a broth, it would have to grow towards the top of the broth in that tube. It has to be near the oxygen. It requires oxygen. It only will grow if there is oxygen, and that's why it's at the top of that broth in that test tube. When you look at B, it's an obligate anaerobe. That means it requires no oxygen, none at all. It cannot grow towards the top of that tube. It has to grow at the bottom of the test tube in that broth where there is no oxygen present. It's an obligate anaerobe. Now, if you look at the facultative anaerobes, which is C, remember they can grow where there's oxygen and where there's not. So you might see more growth at the top. Remember, it does prefer oxygen. So you might see heavier growth at the top, but you will also see it throughout the test tube toward the bottom. An aerotolerant anaerobe will really be dispersed throughout that broth in that test tube. You can see that it's dispersed where there's oxygen and where there isn't oxygen. It tolerates it. Now, the microaerophile, if you look at that test tube, it's a little bit below the surface. It's not at the top of the surface of that broth because it doesn't need a lot of oxygen. It just needs a little. So it will kind of grow towards the top, not completely at the top, but it will be towards the top and definitely not at the bottom because it requires a little bit of that oxygen. Specific types of nutrients can be selected and introduced to different media, like different types of agar, or different types of broth, or different types of liquid material, and we can see if certain bacteria will grow in those conditions. We can introduce a high concentration of salt to an agar and see if certain bacteria can tolerate that high concentration of salt or not. We can introduce them to a sucrose and see if they tolerate that and if they use that, if they have the enzymes to metabolize that. We can use all kinds of different types of nutrients in different types of media to select for optimal bacterial growth. We can use that to identify bacteria based on whether they can metabolize it, based on whether they can grow on it or in it. There are many ways that we can take advantage of bacteria and their nutrient needs. Two definitions that you need to know are inoculation, which means the introduction of microbes into a medium. So a medium is like the agar or the broth. So we can make a streak plate on an agar and we can inoculate it with microbes by streaking it. Or we can use a pipette and introduce bacteria to a broth or to a sugar tube to see if it ferments the sugar. Now, culture is the microbes growing in or on a medium. So we are culturing the bacteria after we introduce them to the medium. Then we hope that they grow in or on that medium. Let's talk about agar. There are all kinds of different types of agar. We use TSA agar. It's a trypticase soy agar. It's kind of like a general agar that a lot of bacteria like. The nutrients in there are kind of broad, and so lots of bacteria will grow on it. There is blood agar that certain types of bacteria can metabolize. There is salty agars like mannitol. Mannitol has 7.5% NaCl. Some bacteria like staph really like that. There are bile salt agars like McConkey. There are many types of nutrient agars, specific agars that have had different types of nutrients introduced into that agar to try and grow specific bacteria or to inhibit specific bacteria. It starts out as a complex polysaccharide. It's a jelly type material. It's not solidly hard and it's not really rubbery. It does have pretty soft consistency to it. You can gouge it. It's not mushy, but it has a really jello-like consistency. Agar, the specific part, that agar part, is not metabolized by microbes. What is metabolized by the microbes is what you put in that agar. So you have this chemically defined media. It's like a recipe. It has an exact chemical composition. You know how much salt is in it. You know how much blood is in it. You know how much bile is in it. You know what's in there. It is an exact chemical composition. It can be complex. It can be a whole combination of things, of extracts and yeasts and meat and plants. All kinds of different things can be put into an agar. And it could be a broth. Nutrient broths have many different types of things in them. And we, again, want to see if that bacteria will grow in it. And then in the broth, we can see also if it's an aerobic obligate aerobe or if it grows anaerobically, like an obligate anaerobe. Enrichment media encourages the growth of specific microbes. That's like McConkey. McConkey wants to only encourage the growth of gram-negative bacteria, and it inhibits the growth of gram-positive bacteria. You can develop a media that looks for a certain type of bacteria down to a specific thing like Staphylococcus epidermis. You can look for that, or Staphylococcus aureus. You can develop a media that grows only that and does not allow anything else to grow. And how the bacteria metabolizes that media that you develop with that specific recipe, with that specific composition, you can see and identify what it is based on whether it grows or not. So enrichment media is a means of isolating microorganisms with specific characteristics from heterogeneous populations. So you are looking for a specific thing that will grow on the specific type of media that you make. Something in the media that you've made inhibits the growth of other microbes, but allows the one you're looking for to grow. Ultimately, what we always want in microbiology is a pure culture to examine. And if we can get to that pure culture, we can also measure the growth of that organism. Streak plates are the most common way of getting to that pure culture state. We can take an environmental sample that has quite a few things going on, maybe a lot of microorganisms, and thin it all the way down to a pure culture, to an isolated colony. Once we get to that stage, then we can do a direct microscopic count. That's where you look at it and you count them. That is just a direct method. You have to count how many colonies there are. It usually requires high bacterial densities, a lot. But the problem with this type of counting is that you usually can't distinguish between living and dead cells. The best way for measuring growth is through serial dilution. And we are going to talk about both of these on upcoming slides. On this streak plate image, you can see the isolated colonies. You can see that it has become a pure culture. You started out with a lot of bacteria and you worked your way down. You made it thinner and thinner and thinner with each pass. And then ultimately the goal, which you achieved, is to get that separated colony. And those separated, isolated colonies are pure colonies. And you can count them. Now, sometimes you might have a whole bunch of little isolated colonies, but you still want to count them with the direct method. You can use a grid like this in order to kind of help you keep track of where those colonies are and where you counted them. A grid is really helpful if you are doing a direct microscopic count of the individual pure colonies. A serial dilution is the best way to get an estimate of the number of colonies in a microbial growth sample, especially because some of the samples have so many colonies that there's no way you could directly count them, even if you had a grid. So you can dilute your sample down, and you take a series of test tubes and you dilute them by 10. Each tube is diluted by 10. So you start out with a sample and you add 1 milliliter to 9 milliliters of this dilution solution. And then that will have been diluted by 10. Then you can take a milliliter of that solution and put it into 9 milliliters of some more of that dilution solution, and you will have multiplied that and it will be diluted by 100. So it's diluted by 10 in each test tube. So ultimately, let's look at the test tube with 10,000. So you've diluted it down to a 1 to 10,000 ratio. You put that dilution onto an agar plate and you count 50 colonies. You take that number of colonies and you multiply it by that ratio of that dilution, which is 10,000. So you can estimate that you had 500,000 colonies in your original sample. So you take that number again of colonies that you grow from that diluted sample and you multiply it by that dilution number. This is a very important concept that you will need for quizzes, exams, and for your homework. It's the microbial growth curve. This is what happens to the microbe after you inoculate it into a medium. After you inoculate it into an agar or a broth, it goes through this growth curve, and it is always this type of growth curve. It has the following phases that you need to know. The first phase is the lag phase. It's the time of gearing up for the division, following the change in culture conditions. What that means is there's this period of time after you've introduced this microbe to the medium where it's kind of just trying to figure out what just happened. It has just been introduced to something. It's been taken from its original environment and put somewhere else. So it's kind of a time of readjusting, of gearing up for replication and growth. Then comes that log or exponential phase. That's the division that occurs of that bacteria, that replication that occurs at a constant exponential rate. You will see on the next slide how that looks. The stationary phase is when the birth rate of that microbe equals the death rate. It's like a plateau. The death phase is a constant exponential period of death. It's a constant exponential death rate. Then you have the phase of prolonged decline. It's the slowed down death rate. Sometimes when you see a microbial growth curve, it will not include this phase of prolonged decline. You can, on quizzes, exams, homework, you can do the 4 phases or the 5 phases. So here is a really good image of that growth curve, and it has 4 phases. That first phase is that lag phase. It's when you have just inoculated the media with this bacteria. There's no increase in the number of the bacteria cells. It's been introduced. It's trying to figure out what's happening. It's changed from one environment to the next. It's getting ready to grow. That log phase is the exponential increase in the number of living bacterial cells. Remember, when we look at growth, we're looking at the number of cells. That's the second phase, the log phase. The third phase is that stationary phase. It's like a plateau, when the birth rate of those cells equals the death rate of those cells. The fourth phase is that death or decline phase. It's an exponential decrease in the number of living bacterial cells. So bacteria goes through this microbial growth curve. It has all those four phases, and you need to know them all, and you need to know what is happening in each of these phases. Prokaryotes can replicate in a variety of ways. For this class, we are going to focus on binary fission, and I'm going to talk about that on the next slide. So in the simplest terms, because this is not a genetics of microbes class, this is just a basic microbiology class. We're just going to talk about binary fission in just a basic way. So what it is the microbe makes an exact copy of its DNA. And then it splits into two separate cells, two separate identical cells. It's just a copy and paste. It duplicates its DNA and makes a copy of itself. Biofilms are polysaccharide encased communities of bacteria where they live together, they communicate with each other, and they protect each other to develop this community. This community can then attach to environmental surfaces. Some examples that you might know of are like slippery, wet rocks when you're playing in a river or crossing a creek. That is an encased community of microbes that are working together and had developed that film on that rock. Pond scum is another example of a biofilm. They are microbes that are living together in a community on the surface of the pond. Your teeth can have a biofilm on there. If you haven't had your teeth cleaned for a very long period of time, you might develop dental tartar. That is a biofilm. Have you ever noticed after you've had your teeth cleaned how different it feels? It's because that biofilm has been removed. It's estimated that 65% of human infections involve biofilms. It's because these communities can work together to become stronger. They protect the organisms from disinfectants and from antibiotics. They work together and encase themselves so that it's really hard for these disinfectants and these antibiotics to penetrate. A very well-known type of biofilm can occur on a urinary catheter. A lot of people may develop UTIs because their catheters are not removed when they should be. They need to be removed in a shorter amount of time so biofilms do not accumulate on those catheters. We have new guidelines where we want to get our patients up and walking and their catheters removed as quickly as possible so these biofilms do not accumulate and they do not develop UTIs. Biofilms can accumulate on all kinds of different non-sterile medical equipment that comes in contact with a patient. And because these biofilms have been linked to so many different types of infections, like urinary tract infections, we need to be very aware of them. Here is a really good image of a biofilm. You can see how it's developed this kind of slimy, sticky kind of community of bacteria. And you could imagine how it would be difficult for disinfectants or antibiotics to penetrate this sticky, slimy mass of bacteria that have formed this polysaccharide encased community. They really are very effective at communicating with each other and protecting each other in these communities. Storing pure cultures is really an important part of microbiology. We need to be able to store them properly so that we can use them for research and to examine them and to identify bacteria and classify them. We have to initially store them. Stored pure cultures are often called stock cultures. In our labs, we get stock cultures in the mail, and then we use those stock cultures to inoculate other media to continue that line of that culture. Stock cultures can come in different forms. They can be frozen in glycerol. You can't freeze them in water because of the ice crystals that can form it in water. But if you freeze them in glycerol, then those crystals don't form. You can also freeze dry them. You can kind of turn them into a type of powder, and they can be stored that way. Now you can store bacteria on agar slants or on agar plates, but this is very short-term storage. You can imagine that it would be short-term because eventually that agar can dry up and it would not be a good environment for that bacteria. You can also store them in broths, and we oftentimes will store our stock cultures in broth, but this is also a short-term storage because imagine those bacteria are living and growing in that broth and then they use up all their nutrients and then they will start to die. Remember, they will follow that growth curve. And unfortunately for both the agar slants and the broths, that microbial growth curve is pretty short. It's for a short-term storage. Death can occur after all the nutrients are used up. Here are some review questions. Which of the following associations is false? A, carbon is used for energy. B, phosphorus is used by microbes for their membranes and for ATP. C, nitrogen is used for protein synthesis. D, sulfur is used for genetic material and membranes. The answer is D. Sulfur is not used for their genetic material and their membranes. Remember, carbon is used for energy. Phosphorus is used for ATP, that energy source, and for their membranes. Remember, phospholipid membranes, ATP, there's that P in there for phosphorus. Nitrogen absolutely has to do with protein synthesis as well as sulfur. Sulfur is involved in protein synthesis, in protein metabolism. So that association between sulfur and genetic material and the membranes is false. The best way to define facultative is A, a microbe requires the condition to live. B, a microbe can tolerate the condition. C, a microbe is destroyed by the condition. The answer is facultative is B. A microbe can tolerate the condition. Remember, obligate is the one that requires something. A obligate aerobe requires oxygen, but a facultative aerobe just tolerates oxygen. It is okay in oxygen, it's okay not in oxygen. Facultative means it tolerates something. A biofilm, A, increases pathogenicity, B, decreases pathogenicity, C, has no effect on pathogenicity. If we think about a biofilm being this polysaccharide encased community, it's protecting each other, they're communicating with each other so that they can resist antibiotics, they can resist disinfectants, they can create these slippery, slimy communities on things like urinary catheters. The answer is A. It increases pathogenicity. And that's the end for Chapter 9, Microbial Growth.